I know that the thing that is on your mind the most is the exam, and that will be happening on Tuesday. I wanted to make a reminder that the way that I structure the exams is to allow you to take it any time throughout the day on Tuesday. I generally try to arrive with bells on at 7:30 a.m. If you would like to meet me bright and early at 7:30 and begin your exam, feel free to do so. If for some reason you can't find me and you know that I'm in the lab, just knock on the door, because it's, I'm there. I'm just maybe in the back hallway or something. So begin as early as 7:30 a.m. and end as late as 9:30 p.m. At about 9:30 p.m., I sort of start pacing around and saying, it's been a pretty long day, can you finish up? And I'm not going to kick you out, but I am definitely going to ask for your respect for me in just recognizing the length of that day and trying to finish off by 9:30. If you have classes all day, and I've talked to one or two of you who simply have classes all the way from 7 a.m. to 9 p.m., and you simply cannot take the exam during the Tuesday that it's offered, let me know, and we will schedule for Wednesday or something along that line. So again, try to come and pick a window any time throughout the day. If you give yourself about 2 hours, you'll be sure to give yourself enough time to finish the exam. While I can say that some people finish it as quickly as 40 minutes, I do have students that linger, and you are welcome to linger, right? I'm not by any means going to kick you out of there until it does get to be about 9:30 p.m. I will just let you keep on working no matter what, and you can finish up in your time, whatever that time frame is for you, okay? Any questions on format of the exam or just general policy on how to take it? I will do 2 review sessions. Those review sessions will be from 2:30 to 3:30 p.m. on Monday in the lab next door to our lab. And then I will also do a night review from 7:30 to 8:30 p.m. on YO courses, on the conferences tool. And when I do that, as I do for my office hours, I will hit the record button. That will record our session so that when you do later go to conferences, you will find that session in a recorded version. It generally does take YO courses a little while to buffer that and get it ready for you to review. I don't know exactly how long that is, but it should be available as soon as that buffering takes place. So any thoughts on that? Yeah. So yeah, the lab. You come to the lab, and that's where I am all day. And that actually is such a great question just in general. I am always in the lab, 7:30 a.m. to 5:30 p.m. on Tuesdays and Thursdays. There are lab sessions that go all day. So that's where you'll always find me on those days. And you're always welcome to hunt me down. And regardless of how busy I appear, just pull me aside and say, Rachel, can I ask you a quick question? I am always more than amenable to answering your questions during that time. I'm just also handling lab. So, I will then take you back and give you an exam and take you into the lab, one of the labs next door, and you can do your exam. Chill. Yeah. 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 So we have lab on Tuesday. Yeah. Yep. Yeah. We just keep right on chugging. So yeah, nothing kind of stops. We just keep on going. But you'll want to take it sometime during that day. Gram stain report in lab is coming up, so that'll be February the 24th. But that's a week from the time that you take the exam. So you still have quite a window of time to think about that lab assignment. The other thing that I would like you to simultaneously be thinking about is your topic for the poster. Some of you feel really, really happy with where you are with your group on the topic and have already emailed me. Some of you are a little less certain about where that topic is. And if you just give me a broad idea of where you are, that's all I care about. My goal for you for the poster is just to help facilitate constant movement. So if I can tell your group is consistently making progress, that's what I'm going to be looking for. I was talking about with Trey this morning, there's so many exciting topics, and just trying to wheedle down now that his group has decided to look at the link between obesity and microbiota and the distribution of microbiota. We know that lean individuals have a particular footprint of microbiota versus obese individuals. And if you take lean microbiota from a lean animal into an obese animal, the obese animal becomes lean. And so that's a pretty powerful and interesting facet of the human microbiome. But of course, there's also the facet of the human microbiome that we understand that the distribution of the microbiome affects your mental health. Both things like depression and anxiety are impacted by the human microbiome. So clearly there's so many subfacets. So if you email me with a topic of human microbiome, I'm going to be like, sweet, that is such a great topic. But then we know we've got our work cut out for us in narrowing that down. And that's what the hypothesis is going to allow us to do. And that'll subsequently follow the topic, right? So there's just a lot that we can consider, and by no means do I expect you yet to be experts on your topic. I just expect you to be making forward progress on it. Any thoughts or questions on that either, or any of the things really that are on our agenda for up and coming? Let me know if midway through the lecture something hits you. And it's Friday, right? So, you know, feel free to scream out, it's Friday, just randomly, if it happens to hit you throughout the lecture. I'm always up for that. It's Friday. So we have done a very thorough job of covering the composition of a prokaryotic, or even more explicitly, a bacterial cell. Right? Norbert, our good pal, was a bacterium, and many of you have emailed me with wonderful answers to the extra credit. Remember that Norbert was a bacillus. So that's actually the simple answer to that question of how we could say for sure that it wasn't Staphylococcus, is that it is a rod rather than a coccus. So that's sort of that simple answer there. But now we need to turn our attention away from the bacterial and archaeal world to momentarily remind ourselves that not all microorganisms are quote-unquote prokaryotes. That is, not all microorganisms are archaea or bacteria. Despite the fact that bacteria are by far the most vast in number, we do have fungi, right? And protists. Many of you saw those on your pond water samples on the first day of lab. So our eukaryotic microorganisms are another important consideration. And in this picture, you just sort of get the idea of how diverse the eukaryotic world is, ranging from what a few of you saw cheek cells on your tooth scraping that you did that first day. And that's a cheek cell up in the upper right. Obviously, that's a eukaryotic cell of a multicellular organism. But then lower right, that's a yeast cell. And yeast, like bacteria, have a cell wall. Their cell wall, of course, does not contain peptidoglycan. Right, good. So we know that it's going to have other structural components. Lower left, those are diatoms. Many of you also saw those on your slides from the Winogradsky columns. They have a silicon cell wall. So again, no peptidoglycan, but another way to add rigidity to their cell wall. Up in the upper left, that is a paramecium, which is a ciliophoria, which is a life form that is a self-contained single cell that can seek and ingest its own food. It actually uses its cilia to help it do that. So what amazing amount of diversity to then say that we're going to try to summarize some of the characteristics of eukaryotic microorganisms and eukaryotic cells in general. And while we see the cell walls of these two eukaryotic microbes, how does something like this paramecium keep some kind of structural rigidity in its cell? Because it doesn't have any kind of cell wall. Where does it get a little bit of structural rigidity? The cytoskeleton. Good, yeah. So Nathan answers the cytoskeleton. Super important in eukaryotic cells. And momentarily we're going to learn that, in fact, it's not just found in eukaryotic cells. It's been observed in bacterial cells as well. But it is very important in eukaryotic cells. So we will cover it in this unit. So recalling that eukaryotic microorganisms include the fungi and, oh, sorry, that was real trigger happy, the fungi and the protists. Fungi, of course, we know our good friend Saccharomyces cerevisiae that we observed in our smear just the other day. Yeast, baker's yeast, is a great example of a type of fungus. So too are molds. So molds with their filamentous growth that some of you saw on your plates that you had swabbed, Those are another good example of fungi. Protists include the informally named protozoa and algae. So even though phylogenetically those aren't official names, we talk a lot about them. So that fits into that group of protists. What distinguishes a eukaryotic cell? I haven't really given this one a name, but maybe we should go with a slightly more feminine identity for this cell, since Norbert had such a masculine identity. But maybe you guys can help me come up with a name, Florence or something. This cell has some very distinctive characteristics about it. And what is the one thing that you think of right away as making this cell different than Norbert? Yes, good. A true membrane-bound nucleus. In fact, this nucleus is surrounded by an entire nuclear envelope. So that is very different than the nucleoid that Norbert had. The nucleoid was not membrane-bound, and that is a big difference. What is another clear distinguishing characteristic of Florence here? Yeah, good. Membrane-bound organelles. Those are things like mitochondria, right, that are the powerhouses of this eukaryotic cell. So we have true membrane-bound organelles and a true membrane-bound nucleus that distinguishes a eukaryotic cell. So let's talk about the cytoskeleton, as Nathan drew our attention to first, noting that this is an important characteristic of eukaryotic cells. It adds that structure and rigidity and shape to the cell, but it also simultaneously plays a role in movement. We're going to begin with the largest cytoskeletal unit, and that is the microtubule. Microtubules are some 25 nanometers across, so they are quite large, and they are comprised of a protein called tubulin. This tubulin protein will orient itself helically, and you can see in this image how that forms a hollow cord structure. It's also interesting to note that the protein tubulin that comprises this microtubule is made of two subunits called alpha tubulin and beta tubulin. Now, does anybody want to help remember what that means regarding the level of protein structure that a microtubule has if we see that this has two polypeptide chains, alpha tubulin and beta tubulin? What level of structure do we know then that this protein has? Quaternary, good, because two separate polypeptide chains interact with one another to form the overall structure. Nice job. Now, there's a lot of functional roles that a microtubule plays. They can play a role in cellular replication, in mitosis. They can play a role in formation of structures called axopodia that we see in buoyant marine microorganisms. These axopodia form these long filamentous projections that allow these protists to float in marine environments. So there's a wide range of roles that a microtubule can play. They also form the central structure of both cilia and eukaryotic flagella. Remember, eukaryotic flagella are going to be different than prokaryotic flagella. Lastly, they also serve as the highways of a eukaryotic cell. What I mean by that is that vesicles quite literally transfer along the microtubules, destined for whatever location their cargo is supposed to go. So I often think of eukaryotic cells as being kind of like a cell city, a city that never sleeps, and the highways of the cell are comprised of microtubules. Lastly, of course, we know that these are playing a big role in shape maintenance. And that, as you all know, if form fits function or form enables function, just the way that a protein would fold also dictates what it does. We know then that if a cell lacks its microtubules, it also will probably lose a lot of its function. This has been studied in nerve cells quite a bit, where if you can create a mutant nerve cell that lacks its microtubules, it will wander aimlessly, forgetting its function. So form, again, fitting function, and very much cytoskeleton playing a role in form. Let's look now at the smallest of the cytoskeleton units. This is a microfilament, can be less than 5 times as large as a microtubule. Notice it can be about 4 to 7 nanometers across. It's just made up of the protein actin, and you can see how it kind of twists around 2 filaments of actin. So this is very small compared to a microtubule, a long twisted polymer. And it plays a big role in motion. And in fact, it's actin polymerization and depolymerization that allows the pseudopodia of amoeba to form. And this is a cool video that I have of an amoeba trying to engulf some bacterial prey. And so you're going to see it here trying to send out 2 of these what are kind of technically mean false feet, which is pseudopodia. And inside of the pseudopodia, you'll notice that the streaming cytoplasm, and that is enabled by the polymerization of actin in microfilaments. So let's just take a quick view of these pseudopodia reaching out and trying to grab the prey. And you can see the streaming cytoplasm. I mean, let's do it again. Beautiful. So think about the fact that not only do amoeba do this in order to capture prey, but also in our bodies, our immune system, what is a cell that does this to? Phagocytosis, right? White blood cells, good, yeah. So for example, macrophages, they're going to reach out and try to degrade any kind of foreign invader, right? They're frontline soldiers of our immune system. And so it's really interesting to note that there are actually certain bacteria that have exploited the actin polymerization chain. So they literally ride on it. Listeria monocytogenes, have you guys heard of that? Have you heard of listeriosis? If you were alive in the fall of 2011, you have. Because remember Rocky Ford cantaloupe? They had this huge scare where people ate infected cantaloupe and they came down with listeriosis. It was a huge deal. I mean, it was especially to me because my hometown, Leadville, is not that far from Rocky Ford, and we used to have track meets down there. And so it was crazy to see like this incredible contamination of their cantaloupes. And listeriosis can be particularly detrimental to those who are immunocompromised. And so it was a pretty big deal. So here's what Listeria monocytogenes does. It gets into our body, and it literally sends out a signaling molecule, forcing our cells to make actin, and then Listeria, the bacterial pathogen, rides on the actin as it polymerizes. That's how it gets around. So it actually forces our body to make it a little train of actin that it can ride. Jake? Yeah, absolutely. So what you're seeing is a long, you just want me to replay it, don't you? So you're seeing the streaming cytoplasm and you're seeing the pseudopodia. There's the one pseudopode that you can really see elongate. The other one's elongating as well. So it's reaching out to try to grab. It's phagocytosis that you're seeing happening there. Yeah. You seeing it? Yeah, cool. Yeah, it's harder to see, you bet. So our third and final cytoskeletal component is intermediate filaments, as you might guess, right? If we've got the small ones and we've got the large ones, here's the intermediate ones. And intermediate filaments, their function is less well articulated. However, we do know that they have a structure that partially includes keratin, which is a protein you probably recognize. Where do we have keratin in our body? Fingernails, hair, right? It's a really strong protein. And so you can see why it might comprise a very rigid kind of structural molecule within a cell. So it's very stable, very supportive. And if you were to look at the ultrastructure, you would say, wow, that looks kind of like a rope, which is fitting. So as we move our way away then from the cytoskeleton, we can journey into the remainder of the cell and start to think about functions of other cellular components. But first I want to ask you a question, and one that maybe will challenge some of your paradigms. This was something that happened in 2001, and I actually have the article up here if you're interested. It was a research group at Oxford in the UK, and what they discovered was two actin-like proteins called MREB and MB1, and they discovered it in the bacterium Bacillus subtilis, which, by the way, is some of you have that as your unknown, right? Bacillus subtilis. And so they're like, whoa, hang on a minute, look at this. These proteins, MREB and MB1, they're actin-like proteins. What does this seem to indicate about bacteria, such as Bacillus subtilis? What portion of the cytoskeleton do these bacteria appear to have? Yeah, good, microfilaments, because those are the ones that are made up of actin. So our answer there to that question is A, microfilaments. But maybe what I want you to take home from this is that if you've ever learned that only eukaryotes have cytoskeletons, that's a false generalization, right? That we actually are seeing at least cytoskeletal components even within the bacterial world. So of course, right, there's exceptions to every generalization that we might give. So into the nucleus that all of you mentioned as being a quintessential part of Florence here, and seeing that it is bound by not just one membrane, but two. Together they form what's called the nuclear envelope. So we have both an inner and an outer nuclear membrane. Interestingly though, the inner and outer nuclear membrane fuse at certain locations, and at those locations there's actually a large pore construct that's comprised of a lot of transmembrane proteins that actually make a pore that is very, very large. It makes porin proteins that we met on the outer membrane of bacteria look like child's play in terms of size. These are very large. They actually will allow fully formed ribosomal subunits to move in and out. So you can see that they're not super exclusive, and what we might say is that the nuclear envelope as a whole is got a lot of high traffic across it. Unlike the cytoplasmic membrane, it's not super exclusive. So the nuclear pore is a transport route, enabling a lot of molecules to make their way in and out of the nucleus. And you could kind of ponder, if you will, different functions of molecules that might be going in and out of the nucleus. And maybe ponder their role in gene expression regulation. There actually are hormones that go in and out of the nucleus that impact gene expression depending on the system. So there's a lot of high traffic. Think about this as like super busy viaducts in our cell city, where a lot of things are merging traffic in and out of the nucleus. So just subsurface to kind of give some structure to the nuclear envelope is something called the nuclear lamina. And speaking of the components of the cytoskeleton, here is a place where we do see intermediate filaments. Intermediate filaments appear to play a big role in allowing the structure that's given to that nuclear envelope through the nuclear lamina. This region here, that's kind of a discrete region, it's not membrane bound, but it's really noticeable. It's something termed the nucleolus. This is a super busy site in our cell city, where in fact it is the location of ribosomal synthesis and ribosomal assembly. So distinct but not membrane bound, cells may contain from one to many, and it is the site of RNA synthesis, site of partial assembly of ribosomes. So let me give you a brain teaser. In a cell that is dividing, actively dividing, why might the nucleolus appear to go away? It is a brain teaser. Because think about what it's doing, Jordan.
It's actively dividing by then, so it should have already synthesized all those proteins.
Cool. Yeah, good. So the focus goes from general protein synthesis to replication. And so I love the way that you put it, in that the focus of the cell, right, shifts to just dividing. And instead of protein maintenance and synthesis, that's something that it's no longer focused on or emphasizing. And so in many cases, the nucleolus will basically disappear in a dividing cell. So we also see in here a lot of what looks like scratchy stuff, but we know that that is what? Yeah, good. It's DNA. I mean, basically you are seeing chromatin there, and they might be at times chromosomes. In the nucleus we're showing, really, they're probably not chromosomes at this point. They're just relaxed, chilled out, because it's a non-dividing cell. We're seeing the nucleolus really clearly. So it is chromatin. And chromatin can take two very different forms, either euchromatin, where the DNA really is relaxed and the genes are very actively expressed. So think this through with me for a minute. In really relaxed DNA, it means that it's not super uber tightly packaged, right? It's not all wound up and wrapped up. And so the genes that are in the DNA in euchromatin, those genes are really accessible. They're like, here I am, come to me, machinery that reads me, right? So they're ready to be expressed. So euchromatin is what we see in a cell that is actively expressing a lot of genes. Heterochromatin is more like what we see when chromosomes form, where everything is really tightly wrapped and gene expression is not really emphasized. But this also has a super sexy-minded other application. And that is what we finally started to realize in the right around early 2000s, late 1990s, is that there was something going on with whether or not a gene was getting recognized and expressed under certain environmental conditions. And that it was based upon whether or not that gene was tightly packaged and unavailable, or whether it was open and ready to be read, right? And this was something called the accessibility hypothesis. And it gained more and more support, that the idea of accessibility, right? Is a gene open and ready to be expressed or not? Or is it hidden and cowered and covered due to packaging? And then this was the rise of what sexy-minded new thing? Epigenetics. Excellent. So if you'd like, this morning I was dorking out with a couple of the early articles, not early, right? I mean, it was 2000s, early 2000s, and thinking about what led researchers first to the accessibility hypothesis and then finally to this epigenetic argument, and how much that has evolved over just the past 10 years, really. So a very cool idea, very, very cool idea. So let's look at the base level packaging of chromatin. And in order to do that, I have made a cheesy model, but I think it is really effective. So if DNA were completely elongated, right, it was just all stretched out, we know that it wouldn't even fit into a cell. It would be, remember how we talked about if we took Norbert's DNA even and elongated it, it would be 1,000 times longer than the cell? And then think about a eukaryotic cell where we've got multiple chromosomes, and there's just a whole lot of string going on there. And so in order to package it, the first line of packaging of DNA, and I think it was Jordan who reminded us this the other day, is around histones. And so this model, I hope, this is like one day out in my garden, and I like hated this old garden hose, and I'm like, you're going to be a histone. And so my garden hose bit the dust and became a histone. But what I've tried to show is that DNA wraps around these histone proteins, and it does so 1 3/4 times. 146 base pairs of DNA wrap around each histone, leaving 54 base pairs of DNA as what we call linker DNA. So this is the core packaging strategy for eukaryotic DNA. And of course, all of you remember that histones are rich in which amino acids? Histidine, okay. And the highly basic, so work with me. Yes, what are some of those basic amino acids? Arginine and lysine, good. So arginine and lysine are going to be very rich in these histones because, of course, they, as Nathan said, have positively charged amino acids. Those positively charged amino acids are like, woo, hello, negative DNA, and wrap around me, right? Feels cheeky. And so that is what happens, right, is that negatively charged DNA wraps around those histones 1 3/4 times, leaving just that linker DNA. And this is often called beads on a string. That is sort of the name given to the very basal level of packaging of DNA. So this isn't it, right? This is, okay, so this is euchromatin, right? Real relaxed, you know? There's just not much packaging going on there. But further packaging can take place. And in fact, it's interesting to point your attention to, there are little proteins called H1. They're histones as well. And they are associated with the linker DNA, so that 54 base pairs of linker DNA. The next level of packaging in DNA is for those H1s to come together. And so if there's an H1 associated with each linker region of DNA, you start to see this coming closer and tighter and being more and more packaged. And this creates something called the 30 nanometer chromatin fiber, or the solenoid. And that's the next level of DNA packaging. Of course, that's not it, right? It can package more from there, and eventually we get that massive condensation that would form chromosomes. Okay? Does that make sense? Like from lowest to highest. And so this is what I'm showing here is actually the beads on a string model of how you would see the base level of chromatin packaging taking place. This is the histone octamer, rich in lysine and arginine, maybe histidine, Jake, right? Because remember, it's the one that can kind of go basic or not. And then we've got histone H1 associated with the linker DNA. And then we have yet another N term. You might have noted that there were a lot of N terms we just went through: nucleolus, nuclear lamina, nuclear envelope, nuclear pore, and now we have the nucleosome. The nucleosome is, by definition, the histone octamer, meaning 8 histone proteins rich in lysine and arginine, the 146 base pairs of DNA that wrap around it, the 54 base pairs of linker DNA, and histone H1. Together, that constitutes the nucleosome. And we could say that the nucleosome is the core packaging unit of DNA. Shall I say that once again? Yeah, okay. So the nucleosome is defined as the histone octamer, the DNA that wraps around it, which is about 146 base pairs of DNA, and then the linker DNA, 54 base pairs, and histone H1. And together, like, that is the nucleosome. So this, I hope, gives you a sense for this beautiful central nucleus, which I think I'm going to pass around this model because I actually really think the way they've done the nucleus is kind of cool looking. So you guys can take a quicker or a closer look to that. So it's interesting to think about in a eukaryotic cell, and this might be something Trey is noticing even as he passes this, is that the nuclear envelope is actually contiguous with the other membranes in the cell. So we actually call that the endomembrane system of a cell. So the nuclear envelope is contiguous with the endoplasmic reticulum. And the rough endoplasmic reticulum is contiguous with the smooth endoplasmic reticulum, which is contiguous with lysosomes, right? So it all is just kind of connected in the cell. So we'll take a look at some of that, but I want to show you just a picture where you can see heterochromatin versus euchromatin. You can also make out the nuclear pore. You can make out the outer and inner nuclear membranes comprising together the nuclear envelope. But then you can kind of see how, oh, wait, here is something that looks like the nuclear envelope, but it's not. It's probably rough endoplasmic reticulum. So you can see how that whole membrane system is just kind of connected. And in this picture, you can see that as well, especially here in these areas. So the endoplasmic reticulum comes in 2 forms, the rough ER and the smooth ER. What does it mean to be rough? It has ribosomes, right? Like, I like to say it's real studly, right? Because it's studded with ribosomes. So these ribosomes literally back up, and it's like they, put on their backup, lights and beeper, and they back up to the endoplasmic reticulum while they're synthesizing protein. So remember, a ribosome's reading RNA, making protein, and once they backed up to the endoplasmic reticulum, they push the protein that they're making into the lumen of the endoplasmic reticulum. So it's literally simultaneously, while the polypeptide chain is being made, it's being siphoned into the endoplasmic reticulum. And then once inside of the ER, that's where it folds. So this is the basic process that is taking place in the rough ER, is that proteins are being synthesized and delivered into the lumen of this organelle. So large flattened membranous sheets called cisternae. We've seen just 2 pictures of those membranous sheets. They appear bumpy because they're studly, because they have ribosomes studding their surface. And it's the site of synthesis of proteins. But these proteins are special. In our cell city analogy, we could think of the endoplasmic reticulum as being the export factory. These are proteins that are going to be exported or inserted into the membrane. So they are destined for some sort of specific delivery. Either they'll maybe be integral membrane proteins that'll grow across the cytoplasmic membrane, or maybe they'll go all the way outside of the cell, where maybe they form scales outside of some protist. So they have some unique destination. They are being somehow exported in some way. So as I said, as the ribosome synthesizes the protein passes through a gate in the ER and into the ER lumen. Lumen just means the inside of this organelle, and that's where they take on their 3D shape. So we might say that as a polypeptide chain enters into the rough endoplasmic reticulum, it begins its process of folding. It goes from being a linear, just primary structure, to taking on its secondary structure, and then its tertiary structure, and maybe its quaternary structure, and then it finally leaves the ER. Maybe a few modifications are made, and then it heads off from the ER. Now, does anybody know where it goes once it's left the endoplasmic reticulum? The Golgi apparatus, yeah. And it's in the Golgi. I think of that as being like the quality control and sort of slight modification factory, where it actually is going to get its modifications, maybe phosphorylation, maybe glycosylation, and then it will be sent to its final destination. We'll get to that in a moment. But first I want to cover what's going on in the rough ER, because clearly that is not involved in protein synthesis. No studs, right? So instead, the macromolecule that gets made in the smooth ER is lipids. Good, good. So remembering back to our macromolecules, that last category of macromolecules. So these are very, very different in morphology as well. They're smaller, they're more tubular, rather than being the cisternae. They look a little more like tubes, though that always organellar morphology differs based upon the particular system you're looking in. But that is a generalization. So they appear smooth because they don't have ribosomes. And as you all told me, they are the site of lipid synthesis. And some of you who are passionate about sports science, and I know a number of you, might also recall that endoplasmic reticulum in a human system actually has multiple different functions. And do you remember the term sarcoplasmic reticulum? And if you haven't studied exercise physics, don't worry about this, but sarcoplasmic reticulum is calcium storage. It's an ER that has that specific function. And it's really cool, actually, to note that there are some protists and some pathogenic fungi that have that too. They actually have like a type of sarcoplasmic reticulum where they do store calcium. So that's kind of cool too. And sterile synthesis, and this is pertinent in the microbial world as well. So whereas we tend to think of, and cholesterol is uniquely animal, so these protists are not making cholesterol, but they do have some unique sterols. And one group of protists that, algae actually, that do this are the dinoflagellates. And the dinoflagellates are the ones that cause red tide, and they cause the ocean to glow. Anybody ever seen that? Yeah, they actually synthesize a lot of sterols within their smooth endoplasmic reticulum, and that's what that reference is for in case you want to nerd out to it. Very cool stuff. So smooth versus rough ER. One of them obviously involved in synthesis of proteins because of its studs. The smooth ER involved more in synthesis of lipids, no studs. So we know, as you mentioned, they bud off of the ER and transport compounds to the Golgi apparatus for further modification and sorting. Quality control center, if you will. So again, just this last note is just a note of that contiguous nature of nuclear membrane, rough ER, smooth ER. It's just kind of all constantly recycling amongst itself and making new from old. And this is a cool picture where you can tell the morphological differences between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. See how this is much more tubular and definitely not studded. And this is a really great shot of the studs, the ribosomes, coating the cisternae of the rough ER. So the ribosomes that stud the rough ER are 80S. How does that differ from bacterial ribosomes? 70S. Why is this very important? And by the way, I've been loving the extra credit answers, but someone who did a super awesome job on the extra credit, tell us why the difference between a eukaryotic ribosome, 80S, versus a 70S ribosome is so important in the world of medicine. Go ahead, Sammy. Mm-hmm. Yeah. Yeah. Mm-hmm. So said, that was a great way to say it. Another way to say it is that antibiotics often target 70S ribosomes. In fact, some of the most famous antibiotics, like gentamicin, streptomycin, erythromycin, the epic Z-Pak, that's like the flavor of the week antibiotic right now, azithromycin is what that's called. They all target 70S ribosomes. So the idea there is that they will kill the bacterial infection inside of you, but not you. And the overarching term there is selective toxicity, right? Selectively killing the bacteria, not killing you. That's actually really epically important, right? We need to be able to, but it's also kind of a fallacy. And I want you to ponder why. Like, why is really 100% selective toxicity not really a thing? So I want you to ponder that as we continue our coverage. But this is an important thing to bear in mind. Who remembers what are the subunits of the 70S ribosome? Not 60 and 40, but good, 30 and 50, good. So the Golgi apparatus, hey, now we've got this vesicle with top secret, cargo that it's taking from the endoplasmic reticulum, and it is delivering it to the Golgi apparatus, right? This precious cargo are the proteins that folded in the endoplasmic reticulum. So now this vesicle is going to merge with one face of the Golgi apparatus, where it's going to then dump in and deliver its protein cargo. Those proteins then will pass through the Golgi, and depending upon their function, they're going to get like their makeup put on, so to speak. They're going to get phosphoryl groups added onto them, or they're going to get carbohydrate groups added onto them. Whoo, loud. Guess we have open lab. So these are like those kind of finishing touches or kind of makeup that you could think of as going onto the protein. So the site of modification of those proteins coming from the ER. Hey, what would we call a protein that got a carbohydrate group added onto it? Glycoprotein. So you might think, oh, interesting, that might become an integral membrane protein that's glycosylated, right? So it might be a glycoprotein. Cool. So vesicles leave the Golgi then at that point to deliver their cargo, and oftentimes it's going outside the cell or into the cell membrane. This is called the biosynthetic secretory pathway. So if we had a protist that is a marine protist, maybe a radiolarian, beautiful, but it has outer external proteinaceous scales on it, would you expect this protist to have a well-formed endoplasmic reticulum? Think that through. Yes. Why? Perfect. So the rough ER and the Golgi are very much involved in secreting the proteins that make the external scales on this protist. So any kind of protist that has an outer shell that contains proteins of some sort, you're going to expect to see a really well-developed rough endoplasmic reticulum and Golgi apparatus. And you know, you could see as many as like 20 stacks of Golgi in any given organism at one time. And there's up to like 4 or 8 cisternate in one stack. So it can be pretty highly concentrated amounts of Golgi apparatus. And I don't, as Borden, as Florence is coming around, does it look like it has a well-developed Golgi and ER? Somewhat. Okay, cool. So let's look at a picture. Here we see the vesicles. You might even want, if you want to, in your notes, you might want to make jot down a memo that these are coming from perhaps the rough endoplasmic reticulum. That might be one place that they are delivering cargo coming from. And so they're going to then arrive at the side of the Golgi apparatus that is termed the cis face. This is also termed the forming face. So from whether these are coming from the rough or smooth ER, carrying their cargo that needs to be modified, they'll merge with the cis face and then be passed through the lumen of this organelle. And the stacks of cisternae that you see here are termed a dictyosome. That's a common descriptive term for that. So then on the opposite side, which you might guess is called the trans face of the Golgi, then the vesicles bud off and maybe they head right to the membrane to insert their glycoprotein that might be cargo in there. So the cis face versus the trans face. Now what's super interesting is that if you happen to become a scientist who is solely interested in the transmembrane proteins of the Golgi apparatus, which could be the rest of your life, right? That could be a thing. So you might be very fascinated in the fact that the cis face, if you were to isolate proteins from that face versus the trans face, you would find that they have a completely different composition, which is crazy cool. So you might say that in fact, the Golgi apparatus is a polar organelle. It actually has a really obvious one side versus another side. It's kind of a cool thing to think about. So I'll leave you with one last note that sacs form on the cis side differ from those on the trans side in terms of thickness, enzyme content, degree of vesicle formation, and I want you to ponder that. And have a wonderful weekend. Please do remember that this is not on the exam, right? So nothing we covered today is on the exam.